Hi, everyone. Today we have as our speaker Dimitar Kostadinov. Dimitar did his PhD with Joshua Sains at Harvard University, working on the concept of self-avoidance. Now, self-avoidance you might think is a cognitive process, but actually it has a lot to do with how dendrites and axons of a single neuron avoid each other. And he worked on the molecular basis of how neurons understand that themselves versus others. Then he joined Mike Heuser's lab at University College London, where he made the beautiful discovery, really important discovery, that the complex spikes, all of our inputs, the Purkinje cells, respond to unexpected reward delivery, omission of reward delivery, and cues that make predictions about future rewards. And he did a really lovely job dissociating these reward signals from movement signals that accompany actions that the animal does. Dimitar is moving on to King's College, starting his own lab in a couple of months, and I can make a strong recommendation for any of you who are interested in his work to contact him and see if he has openings. So Dimitar, thanks you so much for being with us.
Thank you, Reza, for the invitation and that introduction. I think it's really fantastic that our kind of cerebellar community has such an excellent forum in which we can share our work with each other. So really feel privileged to get to present in this series. So today I'm going to tell you about the work that I've been doing over the last several years as a postdoc at UCL in Michael Heuser's lab, where we've been trying to understand neurodynamics generally, and more specifically, the roles of climbing fiber inputs to the cerebellum during goal-directed behavior. So traditionally, when we think about how our brain allows us to kind of execute a behavior, it's like this. We can take some sensory input in this case, of course, the serve coming in from Serena Williams. And if we're lucky, we might think about what this signal may mean, and then use it to generate motor output. In this case, of course, Roger Federer's return. But of course, our own motor actions are going to generate new sensory input. So these actions will actually have a strong influence on our sensory world as well as our thoughts. And we're generally performing these kinds of learned behaviors with some greater goal in mind, like, of course, winning Wimbledon. And of course, the circuits that allow us to perform these kinds of computations are distributed throughout the brain. And in my recent work, I've focused on understanding neurocoding in the cerebellum, a brain region which, of course, needs no introduction here. And I think we can, all probably agree, at least to a certain extent, that this region is kind of intimately involved in closing this loop between sensation, our thoughts, and our actions, and likely does this through its kind of bidirectional communication with other parts of the brain, including the neocortex and the basal ganglia. So most of us probably think about the cerebellum in the context of motor control, and indeed, movements like finger taps evoke strong and focal activation in the cerebellum, but it's becoming increasingly clear, and has been, I think, for a long time now, that the cerebellum is also engaged during a variety of non-motor behaviors. So, for example, in this recent study from Jörn Dietrichson's lab, where human subjects were asked to perform a variety of both motor and cognitive tasks, the authors found broad and overlapping activation in humans by these different types of task parameters. And quite remarkably, I think, the cerebellum is actually probably able to perform these different types of computations using a single canonical circuit architecture. And of course, here is the canonical cerebellar microcircuit that we've known about since the time of Cajal. It gets two distinct sources of input that converge both in Purkinje cells and neurons of the cerebellar nuclei, which constitute, of course, the ultimate output of the cerebellum. And in Purkinje cells, these input pathways drive two distinct types of action potentials. The mossy fiber pathway, of course, is going to modulate the simple spike, sort of the conventional spiking of Purkinje cells, and bring in contextual information from the rest of the brain. Whereas the climbing fiber inputs, which arise from the inferior olive, make these singular and exceptionally strong connections with Purkinje cells and drive complex spikes, which are these bursts of action potentials associated with large dendritic calcium plateaus, and the coincident or non-coincident activation of these input pathways It's going to drive plasticity and learning in the cerebellum. And so I think this kind of circuit architecture and plasticity mechanisms have led to the kind of dominant computational framework through which I, and probably many of you, view the cerebellum, which is that it generates internal models of the world. And so in this framework, inputs from the mossy fiber pathway are going to be recombined based on instructive signals provided by the climbing fiber pathway. And this allows the system to make predictions about the future. And so traditionally, these instructive signals are, of course, thought to carry sensorimotor errors, but as I'll show you in a second, they might carry other kinds of instruction as well. And so importantly, these climbing fiber inputs actually organize the cerebellar circuit. And they do so by projecting to longitudinal stripes of Purkinje cells and form these parallel kind of modular circuits whose Purkinje cell constituents are called microzones. And so these modules are probably the fundamental computational subunits within the cerebellum. And because these modules are defined by the specific projections of climbing fibers, we can think of these inputs as really constraining what a given module can learn, and because they can only learn what they're taught by their climbing fiber inputs. So given the importance of these inputs for learning, this is what we've focused on in our recent work. And we've tried to kind of address the diversity of information that these climbing fiber inputs can bring to the cerebellum. And so today specifically, I'm going to tell you first about our efforts to understand the organization of activity in these instructive climbing fiber inputs during a learned behavior. And then in the second part of the talk, I'll tell you about our more recent work where we've asked how these instructional signals are themselves modified by learning. And finally, I'll just speak very briefly about where we're going next, which is to ask how the cerebellum may actually engage with the rest of the brain during behavior. So to study cerebellar computation during behavior, we've developed this virtual reality-based sensorimotor integration task for our mice to perform. And this was inspired and is somewhat related to the task used by the International Brain Laboratory that some or many of you may be familiar with. So in this task, mice are situated in front of an array of monitors, and we train them to use their forepaws to turn a steering wheel from an eccentric visual position into the visual midline. And if they hit this kind of central target region, they get a reward. And if they undershoot or overshoot the target, that's an incorrect trial and they don't get a reward. And so as you may be able to guess, we kind of designed this task with the cerebellum in mind because it allows us to ultimately impose behavioral perturbations like gain changes and study adaptation. And I'll touch on that later on. And so this is what the behavior looks like in a trained mouse. So just to orient you all, the object is going to appear here on the left-hand side. This cyan region is where they're aiming. And the mouse is actually in this little box here, and you can kind of see its tail kind of flipped up here. And so here, when I play the video, object appears, mouse turns the wheel and gets a reward. And here is one more time, object appears, mouse turns the wheel and gets the reward. And so here's the same thing from a frontal view, and you'll see the mouse doing the task, and I'll just spell out what's happening on the screen here on the right-hand side. And so here the trial begins, mouse turns, it gets the reward and gets its little sugar water from this lick port that's kind of in front of its face. Here it is one more time. Okay, so while our mice are performing this task, then we record postsynaptic climbing fiber signals from Purkinje cell populations. And we do, expressing genetically encoded calcium indicators, and we do this using two-photon imaging. And so here's a Z-stack through a region of the cerebellum, lobule simplex, and as this kind of Z-stack moves down, you see the individual dendrites of Purkinje cells flashing away and the cell bodies associated with those dendrites down below them. And so this is a zoomed out view just to give you kind of a more general perspective, but our imaging fields of view are about half of the size. And so here's one of our actual recordings just sped up for you a couple of times. And we take these movies and we segment them into the regions corresponding to individual Purkinje cell dendrites. and extracted dendritic calcium signals from these Purkinje cells, which have been shown in a number of studies to faithfully report climbing fiber input and complex spiking in Purkinje cells. And so after making these recordings, then we aligned the activity of individual neurons, like in this kind of field of view, which has about 270 neurons, to important behavioral epochs. And the first thing we did was align to movement onset itself. And so here I'm showing you a heat map of the average movement aligned activity in all of the neurons in this particular field of view. And each row corresponds to the activity of a single neuron, and the neurons are just sorted by amplitude. And as you can probably see, many of these climbing fiber inputs are activated just prior to the movement onset. And so this is only on correct trials, but it was also the case on undershoot and overshoot trials. And so to a first pass, we found that these movement predictive signals are actually similar across our different trial outcome conditions. And this actually makes sense if you consider the movement trajectories themselves, which start out quite similar to each other and really only diverge later on in the movement as the animals are kind of adjusting where they're going to put the target at the end. So what was more interesting, though, is what we observed when we aligned to movement offset and reward, which always happen at a fixed delay relative to each other in our task. And so our naive expectation when we acquired these reportings was that we might be able to pick up some signatures of kind of sensorimotor errors on incorrect trials of this task. But instead, what we found was this prominent modulation at the time of reward delivery after the mice had performed the task correctly. And this activity was substantially more prominent than the activity we recorded on incorrect trials, where the animals undershot or overshot the target. And what was even more interesting was that we noticed that this modulation was actually bidirectional. So some cells were activated by the reward, and others were suppressed by it. And we wondered whether this might actually reflect climbing fiber inputs to distinct cerebellar modules. And so just to explain what I mean here, as many of you probably know, neurons in the inferior olive actually don't project randomly into the cerebellum. And instead, neighboring olivary neurons project to adjacent regions in the cerebellar cortex. And moreover, these olivary neurons are actually gap junction coupled to each other. So we can kind of schematize the circuit like this. Neighboring groups of these gap-coupled olivary neurons project to neighboring groups of Purkinje cells in the cerebellar cortex. So the correlated climbing fiber inputs of these gap-coupled olivary neurons are translated into correlated instructive signals and complex spiking in Purkinje cells. So this is what we think about as the kind of anatomical and functional basis for the modular organization of the cerebellum. So now when we took our imaging fields of view and ordered our neurons spatially, we noticed that neighboring neurons actually had similar reward-related activity, alternating between groups that were activated and groups that were suppressed. And so to classify these neurons systematically, we developed a procedure using principal component analysis and clustering to identify Purkinje cell microzones based on their spontaneous complex spiking activity. And we identified these functional clusters, which then we sorted, and this kind of manifests as this canonical block diagonal correlation structure, which has been described previously. And so now when we took these functional groups color-coded by these different colors and mapped them back onto our original imaging field of view, and now remember that this kind of functional clustering didn't have any anatomical information to begin with, we found these beautiful parasagittal stripes corresponding to our microzones. And so this clustering was done purely based on function and not based on anatomy at all. Yet we reconstruct the kind of anatomical stripes based purely on functional correlations. And so now when we looked at what these microzones were doing during our task, we found that Purkinje cells within a given microzone exhibited remarkably similar task activation in response to rewards. And we could classify these microzones as either reward activated, like this guy, or reward suppressed, where the activity actually went below the baseline firing rate of the Purkinje cells. And now when we looked at what happened on incorrect trials, we observed similar activity during the delay period, and only divergence in these kind of responses at the time of reward delivery. And so this is just in a single animal, but this is what our microzonal responses look like across all of our recordings. So here is the mean activity of these two groups when they've been split by activated and suppressed microzones. And when we looked at, as I said, what happened on error trials in our task, we found that these reward suppressed Purkinje cells were preferentially activated on incorrect trials of the task. And importantly, this error-like signal wasn't expressed at the time of the end of the erroneous movement, which happens here, but was in fact present at the time of reward delivery. So the error seemed to be computed based on the reward and not the movement itself. And I should also note that both reward activated and reward suppressed microzones were actually able to signal movement in about equal proportions. Okay, so this finding intrigued us because it was kind of inconsistent with the traditional role that we described to climbing fiber inputs and complex spikes, which is to encode these motor errors. So we kind of explored these reward-related signals further. And we hypothesized that these signals may represent the evaluation of some kind of learned prediction, rather than the motor action per se. And so we tested this directly in a couple of different ways. So the first prediction we made was that expectation should modulate these signals. And so to test this idea, we actually added two types of reward delivery kind of contingencies within our task. And so in the majority of our task trials, things proceeded just as you would expect. But on a subset of trials now, we added two types of extra rewards. The first one was completely unexpected rewards that the animals, couldn't predict acutely. And interestingly, we found that these unexpected rewards evoke significantly larger responses than the expected ones. And the other was a non-trial reward type was fully predictable. So we gave these cued rewards that were preceded by an auditory tone. And in this case, we observed strong activation to the cue itself, but not to the reward. So it's as though in these predictable rewards, which didn't violate the animal's expectations, the reward itself was no longer really a salient stimulus. And so I think together, these kind of recordings demonstrate that increased reward expectation exerts a kind of suppressive effect on the responses triggered by the reward delivery. And in some ways, and quite remarkably, these neurons are behaving much like you might expect a midbrain dopaminergic neuron to do. It's the kind of brain's canonical reward processing. Okay.
May I ask a question about this?
Sure.
When the animal is producing the task and moving the cursor to the reward zone, doesn't the visual feedback provide the cue regarding whether it's going to get rewarded or not?
Yeah, I mean, so I would say that the visual cue does provide that information. Whether and how much the animals are like kind of explicitly paying attention to it is kind of a different question, right? So it could be that it could be that the animals are kind of using the object appearance as a cue to turn the wheel, and not necessarily using that endpoint information. Yeah. So yeah. Does that answer your question?
So in principle, the information is available whether an animal.
That's right. That's right.
Jennifer has raised.
Yeah.
Yeah.
Jennifer.
Hi. This is such a cool task, and I think a similar related question about like what exactly is being coded And how do you know that, or how have you looked at the possibility that it's something like swallowing, like a motor? Because animals do things when they get rewards, and they do different things when they don't get rewards, and they might do different things when the reward is unexpected versus expected. And so this is always a tricky thing, how to disambiguate reward related signals per se from the behaviors, the whole suite of behaviors that goes with reward.
Sure. Yeah, I mean, we did, like, I've kind of skipped over this because it's already, because I didn't want to get into kind of all the details and controls, and I wanted to kind of get past this and show the kind of newer stuff. But we did, we tried to look at how, licking might modulate this. So we split based on trials with lots of licks and trials with few licks, and found that there wasn't really much of a difference in the responses and so on. And I think I don't think I can speak to your question about swallowing specifically. That's not something that we monitored, but in terms of the actual lick rates and lick timing relative to the reward, I think that doesn't seem to play a role here.
Yeah, and I mean, there's motor, and then there's also, again, like Reza was saying, there's sensory correlates of the reward or not reward, like the feel of the water, the
Sure,
the taste
yeah.
of the water. And
Yeah.
Thank you.
and I mean, I think the dopamine people have like have some nice approaches to get at that, right?
So I mean, even in the original, like, in some of the original papers, like changing the like, we haven't gone through and parameterized, all of the things that people in the dopamine literature have done, like changing quantity, predictability, doing blocking experiments, like I think Court showed very nicely a few weeks ago. But I think that kind of getting at all these details is something that we're obviously thinking about going forward.
Thank you. Rich had his hand up. Rich, do you want to ask your question?
Just relevant to your question, I noticed that the event rate's much higher in the reward-activated microzone when the wheel stops, which might be relevant to that question about, is the visual cue actually signaling the likelihood of an upcoming reward?
That's right. So you notice this little purple line is higher, right? Yeah. I think.
No, I'm saying at the when the wheel stops at the cue point, that it's going up to 9 hertz on the left here,
Yeah,
3.5 hertz on the right.
Right. So sorry, I don't think I understood what the.
Oh, well, it seems like, again, there you have a difference at the time, and it may just be this particular case, but there you have a difference in the event rate.
Yeah.
Based on the visual, presumably based on the animal, either the movement itself or the animal seeing that the cue is moved into the reward zone on the left for the reward trials, right, and not on the other trials.
So the visual cue is is shown in purple here, and so the maybe let's I think there's supposed to be questions at the end.
Yeah,
I'm happy to do this, but is it okay if I just keep going for now?
Recommend that, yes.
Yeah,
Thank
great.
you. Yes.
Thank you. All right, cool. So the other type of kind of perturbation that we used was on a subset of correct trials in a task to actually omit reward delivery. And so to illustrate what this actually looked like in a single field of view, then we I'm showing you here kind of the difference in the response between the trials when the animal got its reward for real and the ones where we kind of did everything just as we had on the real trials, but it omitted the reward. And you see this kind of stripe of activation come just after the time of the that the animal expected the reward. And so here, these fictive rewards evoke error signals in both reward activated and suppressed microzones. And we also omitted cued rewards. And here we found that that while the kind of differences were a little bit weaker, omission of cued rewards were present in both types of microzones, but actually preferentially in these reward suppressed Purkinje cells. And so finally, to validate these signals, using electrophysiology, we took Neuropixels probes and which many of you are probably familiar with, and recorded the activity of Purkinje cells using electrophysiology directly to see whether these signals were in fact related to complex spiking directly, or some weird dendritic kind of calcium events in our Purkinje cells that were kind of artifactually related to the imaging. And so here I'm showing you a Neuropixels probe and a recording track that going through lobule simplex. And so here's raw data from one of these recordings. The Purkinje layers in Neuropixels recordings are quite obvious. So you can see these bands of high firing neurons, and some of you, if you squint, probably can actually even pick out some complex spikes in these Purkinje units in this kind of band where there's probably multiple cells being recorded at once. So using these probes, then we could readily identify both a simple and complex spikes from our Purkinje cells, and we were confident that we were recording from well-isolated units because we could observe the kind of canonical post-complex spike pause in the cross-parallelogram between our complex spike and simple spike units. And because this was more of a validation experiment, we trained our mice on a kind of minimal version of our task, where we just gave them cued and random rewards. And indeed, we observed Purkinje cells that were both activated and suppressed by random reward delivery. And this reward modulation went away when the rewards were cued. And so, yeah, so we were able to kind of validate our imaging results again using electrophysiology. Now, more recently, we've been asking how these signals may actually change over the course of learning. So, as I said before, asking if the cerebellar instructive signals are themselves subjects to instruction. And so, to do this, we've been performing longitudinal experiments over the course of learning, following animals as they transition from naive subjects in our task through a learning phase to become experts, and then applying sensorimotor gain changes in our expert mice. And so as our mice were learning, we record, once again, population activity from climbing fiber inputs to Purkinje cells, both in lobule simplex and also now in an adjacent region called lobule 5. And we targeted these two regions because we knew that they were both engaged in forelimb movements in trained animals. And as before, we extract the calcium transients and complex spikes from individual Purkinje cell dendrites in these regions. And so now in these experiments, we return to the same fields of view day after day and align the activity of individual Purkinje cells across these days, allowing us to track the dynamics of these neurons across learning. And so our ability to kind of match individual neurons across time degraded kind of linearly day over day. But this wasn't due to some kind of degradation in imaging quality, because we showed that the firing rates of our individual Purkinje cell dendrites were stable across time, and also our ability to identify kind of firing neurons on any individual day wasn't subject to kind of degradation over time. So having ensured that our preps were kind of healthy and stable, we performed several analyses. And the first was to compare task encoding across these regions in our trained mice. And once again, we began by aligning to movement onset, and here found that the climbing fiber inputs to lobule 5 preferentially signaled movement onset in our trained mice. And conversely, inputs to lobule simplex preferentially signaled trial outcome through this kind of reward-related activation. But of course, this interpretation is a bit confounded by co-occurring task events, like, for example, the object appearance, which is correlated with the animal's initiating movements, and licking, which is associated with the reward delivery. So to tease these things apart, we built generalized linear models where we use task events to predict the neural activity of our individual Purkinje cell dendrites. And as you can see, these models actually do a pretty decent job of predicting the activity that we reported as our animals performing the task. And then we could shuffle individual sets of task events, like the ones related to licking, or the object's position and trial onset, or the wheel movement itself, and kind of movement initiation, and determine whether these sets of task parameters were actually contributing significantly to the firing properties of our recorded neurons. And when we did this, we found that indeed this works. So models with shuffled movement or reward timing degraded the model performance in many of our neurons. And now applying this approach to our whole recorded population, we observed that kind of doing these shuffles degraded our model's predictive power as expected. So here is an example field of view where the movement shuffle ruins the predictive power of our models in lobule 5, and similarly the shuffling the reward timing ruins the predictive power of our model in lobule simplex. And importantly, shuffling other shuffling other task events, like these, like the object related predictors here, and the licking predictors here, didn't degrade our model performance, suggesting that this was kind of a unique, demonstrating kind of the unique contribution of these particular task events. And so we show systematically that lobules 5 and simplex do indeed have complementary encoding for the movements preferentially in 5, and reward-related signals preferentially in lobule simplex. And so now, having established this task encoding in our trained mice, we could then compare this encoding to our naive sessions in the same neural populations to determine how these signals might change with learning. And so here we're going to compare naive to learning to expert sessions, what we're kind of collectively calling slow learning. And so the transition from naive to learning expert sessions is a bit hard to compare, because actually the mice are doing things a little bit differently. And the main difference that we can compare is in terms of engagement. And indeed, once the animals get going and learn how learn the kind of structure of the task, they tend to initiate many more trials in these learning and expert sessions than they do in the initial naive sessions. And here I should say that the naive sessions are kind of grouping together the first couple days of the task, and if we looked only on the very first day of the task, this engagement would be even lower. So what's a bit more interesting, though, is the changes that occur between the learning and expert sessions. And so, yeah, so the difference between these learning and expert sessions is really a matter of kind of proficiency, because here the task structure or the kind of rules that leads to getting a reward or not getting a reward are exactly the same across these conditions. And so in these learning sessions, mice know to make wheel turns for reward, but they're just not amazing at the task. And so their endpoints kind of spray the target area and produce many undershoots and overshoots, with a kind of slight tendency to produce undershoots, which is something that we see consistently across animals. And so their skill level improves over time, and over the course of about a week, the rate of correct trials increases and kind of plateaus. And so now looking at what our climbing fiber inputs were doing in these different sessions, we can see some kind of functional correlates of these differences. And so the first thing that we observed was that predictive signals, like these movement signals that showed in our kind of in the previous part of the talk, actually emerge over the course of learning. So I'm just showing this for lobule 5, but in fact it's also present in lobule simplex. And the second is that predictable task events like trial reward delivery actually are progressively suppressed as our animals are learning the task. And so to quantify these changes systematically, we attempted to fit the climbing fiber activity in naive and kind of learning sessions based on models trained in expert mice and vice versa. So we did kind of various different combinations. And importantly, this is in the exact same matched neurons across learning. And so perhaps unsurprisingly, our generalized linear model performance across sessions wasn't amazing. In particular, models trained on learning and expert sessions did a very poor job of predicting activity in our naive sessions. And interestingly, the models actually did a bit better at predicting activity in the naive sessions in lobule simplex, where reward delivery was more prominently represented, than in lobule 5, where movement anticipation was more prevalent, and really only present on the kind of learning and expert sessions. And so that's actually just shown for you here. So here I'm showing you the kind of fraction of neurons with matched fits based on the different stages of learning. And so you can see that that, for example, a model, the movement signals really only emerge after learning in lobule 5 and in simplex, whereas in lobule 5 and simplex, whereas the kind of reward-related activation, which is present throughout the learning process, is more consistent in simplex. Okay, so now this is learning on a slower time scale, but we can also look at how this learning happens on a fast time scale in single sessions by introducing sensorimotor gain changes. So in these gain change sessions, things start out perfectly normal for our mice. We give about 60 trials at the sensorimotor gain that the animals have been trained on for weeks. And after these baseline trials, then we introduce a gain increase. And so just to show you, just to tell you all the things I'm showing you here, this is kind of a histogram of the endpoints of the mice's trajectories. So they're aiming for the central region. And as you can see in the baseline trials, they're predominantly hitting the target with similar performance to what we had on the expert sessions, because these are kind of oftentimes taken on adjacent days. And here I'm showing you the wheel trajectory, and you can see the animals are kind of again aiming for this central target region, trying to earn their rewards. Now, when we so this is what the mice's behavior looks like just after we introduce the gain change, and as you'd expect, the object, this is a gain increase by a factor of 1.6, and it's designed so now a perfectly aimed movement will systematically produce an overshoot. And of course, this is what happens. The animals start having many more overshoot trials, and you can see here this is where the object ends up when we've applied the gain change, and this is where it would have ended up if we hadn't applied the gain change. So it's very similar to the position that it was in before. And so as our mice are adapting, they kind of go through this intermediate phase, which we define as the block of trials in which they get the fewest correct trials relative to what they would have expected under these baseline conditions. And so this kind of mid-adaptation is where their performance, the performance prediction error is probably the highest. And eventually, though, they end up scaling their movements down, and their performance increases back up. And this happens within about 100 to 120 trials of the initial adaptation that we imposed. And finally, then we change the game back so we can get some kind of washout data. And here in these washout trials, which happen immediately after the game change, we see that the movement is smaller than it was pre-adaptation, just at least for the first kind of set of trials. And so now, when we look at our defined movement coding and trial reward coding neurons and ask whether and how they change their firing patterns during adaptation, the first thing that we can show is that our movement coding neurons actually change very little across these different phases of learning. So we can say that our movement predictive signals are actually consistent during the course of adaptation. But now what about this kind of trial outcome reward-related activity? Well, here we see something more interesting. So after we take our baseline measurements in these neurons, we can then compare the different phases of adaptation that I described to you before. And so at first, after we change the gain, the outcome signals in lobule simplex actually start to increase. And this increase actually continues during this kind of middle phase of adaptation, where, remember, I told you that the actual performance of the mice kind of deviates most from what it would have been if we hadn't imposed this gain change. And as their behavior improves, these signals actually begin to decrease. And finally, when we change the gain back to baseline, we still have some kind of some residual performance error. So the signals actually stay above baseline. So it seems like these reward-related signals, specifically in lobule simplex, are actually scaling transiently during the kind of dynamic phase of the adaptation. And so with that, I'll actually just very briefly summarize what I told you today. First, I told you about our kind of published work showing that climbing fibrams to Purkinje cells encode both sensorimotor signals as well as reward context. And this reward modulation is bidirectional and organized by microzone. And that unexpected rewards evoke stronger modulation than expected ones. And that we observe what look like reward-related error signals in both operant and associative context. And then I told you about our more recent work where we've been trying to understand how these climbing fiber signals actually change over the course of learning on both slow and fast timescales. And we show that during task acquisition, predictive signals emerge and responses to predictable events are suppressed. And that on fast timescales, our operant reward responses actually grow transiently during the dynamic phase of adaptation. And so if I can just kind of give kind of general summary of what we demonstrate, I think it's clear that instructive climbing fiber inputs to Purkinje cells can actually transcend sensorimotor errors and evaluate the outcomes of learned predictions. And we kind of tried to, Mike and I tried to summarize these ideas in a review that we wrote last year, if you guys are interested in this. And I should say that that these ideas aren't just ours and are inspired by probably several decades of research into the non-motor aspects of cerebellar function. And in fact, some of you on this call are the people that inspired us to kind of do this work. And we also had contemporaries in terms of demonstrating these kind of reward-related signals in the cerebellum, including people like Kurt Hull in his lab, and Mark Wagner and Beach and Lowe at Stanford, and also prior work by even Reza's behavioral work in humans showing that you could kind of tease apart sensory prediction errors from reward prediction errors with some really clever behavioral experiments. And so, yeah, building on these ideas, I thought I would just very briefly tell you about where we're going next, with the caveat that these experiments are kind of exciting, but actually very preliminary. And so the first thing I'd like to show you is our efforts to try and map inputs to specific climbing fiber pathways. And to do this, we're employing retrograde transsynaptic rabies tracing, seated in either lobule 5 or simplex. And then we do the complementary injections of the rabies cocktail in the inferior olive, and can then look for inputs to olivary neurons, which are going to project to either simplex or lobule 5. And as you'd expect, we kind of find the canonical regions that we know, or four-brain regions, sorry, that we know project to 5 and simplex, namely the mesodencephalic junction and motor cortex. And now when we look across the brain, we find a variety of regions that project differentially to olivary neurons targeting these two different parts of the cerebellum. But interestingly, what's missing is a direct projection from the canonical dopamine reward system to the inferior olive. So we think that whatever this reward-related activation is in the cerebellum, it's unlikely to be inherited directly from the kind of midbrain dopamine neurons. And now in parallel, we're actually piloting functional experiments to assess how the cerebellum might engage with the forebrain. And so to do this, so to drive activity in specific cerebellar output pathways, we can utilize this combination of imaging and cellular resolution photostimulation, the kind of all optical methods that was pioneered by my postdoctoral laboratory, and combine this with Neuropixels probe recordings in a couple of regions. And so here's an example of this kind of experiment here, where without knowing the specifics of the connectivity, we hedged our bets and looked for coupling between the cerebellum and motor regions of the thalamus and cortex. And here I'm showing you our probe tracks going through motor cortex and thalamus, and an imaging field of view where we've expressed GCAMP to image Purkinje cells and the optogenetic molecule carmine to activate these same neurons. And so in the cerebellum in this experiment, we targeted two distinct groups of Purkinje cells, which correspond to two different microzones. And as you can see, when we play this movie, we're able to simulate them independently. And this is just quantified for you here on the right. And we perform these simulation experiments while recording in cortex and thalamus, and we're able to find neurons that were differentially modulated by activating these neighboring, but probably functionally distinct groups of Purkinje cells. So kind of this, I think this is going to be a fruitful way to then assess how cerebellar modules with particular functional properties might engage parts of the forebrain that may have similar or different functional properties. And so with that, I'll just conclude and thank the various people that contributed to the work that I showed today. So the kind of main parts of the studies that I showed were done in collaboration with a PhD student, Maxime Beau, and a master's student, Marta Blanco Pozo, who's now moved on and has been doing a PhD at Oxford for several years, and also supervised by Beverly Karp and Mike Poyser. And the transsynaptic tracing experiments were done together with Federico Rossi and Sam Clothier, and the all-optical experiments were done with Nick Robinson, Morris Buchholz, and Edgar Baumler. And Henry Delvish, who's a graduated PhD student in the lab, was instrumental in kind of getting this across-day registration pipeline going. And also I'd like to thank Dana Cohen, who's did a sabbatical in our lab, Arndt Roth, and various members of the Poyser Lab Cerebellum team over the years for their kind of support and advice. And finally, I'd like to shamelessly plug the fact that my lab is opening in July, and we have various positions. We have funding for various positions at for technician students as well as postdocs. So if you're interested in the work that you saw today, please feel free to get in touch. And with that, I'm happy to take any questions that you might have.
Thank you, Dimitar. That was wonderful. Yeah, let's open it up. I'm sure there's going to be many questions for you.
Should I keep sharing, or should I just
Well,
maybe I'll keep it up in case people have
yeah, I think you want to keep it up. Yeah. Well, looks like we have a question from Tabal. Tabal, did I pronounce your name correctly? You have a couple of questions on the chat.
Yeah. My name. So I have a couple of questions. The first is related to so lobule 5 and simplex. Did you test whether they are actually essential if you disturb them during learning, how the behavior actually would change? And the second question regarding the fast learning adaptation. You mentioned that there is a high, like, complex spike rate ramps up a bit during like the adaptation phase. Is this related more to the reward like error that happens, or related to the sensory deviation or deviation of prediction of expectation of the visual stimulus being in one place and then shifting?
Right. So so your first question about these regions being essential for learning, we haven't done that experiment directly. I think that it's been well established that these regions are important for forelimb movement. So so in mice, I think the kind of study that first showed that you could actually evoke forelimb movements and even kind of bind sensorimotor associations in using this part of the cerebellum was a paper in 2015 from Tom Otis's lab. Yeah, in a kind of slightly different but related sensorimotor task. Court Hull did a really nice set of experiments demonstrating that if you kind of wash on NVQX or some kind of AMPA receptor blocker, so basically to prevent climbing fiber inputs from exerting their influence on Purkinje cells, and also to prevent the kind of dynamic parallel fiber modulation of Purkinje cell outputs in this same region, you perturb the mouse's ability to kind of adaptively time its forelimb movements. So I think that these regions are well established for their roles in forelimb movements. Now, in terms of your second question about whether this might reflect some visual prediction error, I think that personally that this is unlikely, because the end of the object, the end of the movement in our task happens about 500 milliseconds before the reward is or isn't delivered. And so if we were to see a kind of endpoint error signal, which quite frankly, I've looked for extensively in our task and had very little success in finding, I think that would have happened, more closely timed to the end of the movement rather than at the time of the reward delivery itself.
Thank you so much. Violetta, you have your hand up.
Yes, thank you. And thank you for a nice presentation. One question regarding the last project, the one that tried to trace the areas that could modulate these functions. And I was just thinking there's a more exotic hypothesis on this, on the dopaminergic inputs to cerebellum from the locus coeruleus.
Yeah.
Did you thought about this, or
yeah, that's a really interesting question, and yeah, I think that would be a cool place to look, in particular because the kind of reward-related error signals that we see actually look a lot more like the kind of unsigned prediction errors that you see in locus coeruleus than the kind of signed ones that you see in the dopamine system. So yeah, I think that is actually probably a more yeah, the relationship between the cerebellum and locus coeruleus is potentially a more interesting one to explore in this regard, especially because we know that we know that there actually are connections. In fact, there's a paper showing that Purkinje cells themselves might project directly to the locus coeruleus. So yeah, I think that's a really interesting point, and one that I think we should probably explore further.
I guess to follow that up, did you measure pupil size in any of these experiments?
We have behavioral videos in these experiments, but we haven't looked at it to look at how this might relate to arousal. But yeah, we could absolutely do that.
But any other questions for Dimitar? That was wonderful, Dimitar. Thank you so much for your time. Brilliant work. And thank you, everybody. I hope you have a wonderful day.
Thank you. Take care.
Bye.